My name is Dr. Gail Roboz. I'm a professor of medicine at Weill Medical College in New York City and the New York Presbyterian Hospital. I am the director of the leukemia program there, and I'm speaking to you today from Munich, from the 17th International Acute Leukemias meeting here. I serve on the steering committee of this meeting, and it's a particularly wonderful scientific exchange focused exclusively for several days of intense topics in acute leukemias. My job during this meeting was to talk about novel immunotherapies for AML. And immunotherapy for AML is a very broad topic, actually, which has received quite a bit of coverage during the meeting on specific drugs, but mine was more of an overview topic to see what are the problems potentially with immunotherapies, starting with what's the right target, both with respect to AML and with respect to the immune system. So my lecture actually discusses a number of different emerging and existing targets on AML cells, blasts, and leukemia stem cells that may be relevant for acute leukemia. For example, CD33 is tried and true for decades, but actually, although we have an approved immunotherapy in the form of gemtuzumab ozogamicin, which is a conjugated anti-CD33 antibody, we don't know 100% why this actually works, especially in the core binding factor leukemias, which arguably have less CD33 expression than others. So here we have decades of study for an antibody. It is currently in testing in a number of different formulations. We still don't know exactly why it works, and we've had trouble getting it to work effectively in AML other than for gemtuzumab. Another target is CD123, which is expressed on leukemia stem cells. CD123 is being looked at in antibody format, in dual antibodies like BiTEs, and tandem antibodies, DARTs. There are lots of different forms of antibodies under development, and also being looked at in CAR T-cells. And basically here too, there have been smatterings of responses both with the antibody constructs and with CAR T-cells, but no home run yet. And the question is whether CD123 is actually being successfully targeted on the AML cells, or whether we are being kind of complicated by the on-target but off-tumor side effects. So lots of CRS is being seen, cytokine release syndrome is being seen in assorted immunotherapies, which is a problem for AML development. And also, if you look at, for example, the checkpoint inhibitors in acute leukemias, the inflammatory side effects that our solid tumor colleagues are used to seeing, like nephritis and pneumonitis and some of these other very specific complications related to checkpoint inhibitors, are a problem for AML too. So basically, broadly speaking, for antibody development, for checkpoint inhibitors, and for CAR T cells, we have seen some responses. There are patients with acute myeloid leukemia who do go into at least a partial response, but lots of toxicities, different for the different constructs. And the question is, going forward, how to mitigate the toxicities while increasing the on-target, on-tumor effects. Don't have the answer to that question right away, but I have highlighted a few drugs that are getting looked at. For example, there is a novel CD70 antibody. This is a putative stem cell target that is being investigated in combination with hypomethylating agents. There is a radiolabeled CD45 antibody being looked at in patients with active leukemia as part of their conditioning. And there are lots of different immunotherapies currently in development in phase 1 and phase 2 trials, again with very early data. So that's sort of a broad sweep of some of the problems and issues with immunotherapy that are getting attention at the Acute Leukemias meeting.